And George, since you're no longer with A&M, can we still say howdy? Is that prohibited?
Yeah, no.
We can still say, okay. Well, howdy to the Aggies at least.
Howdy.
So this morning, as George mentioned, what we'd like to do is share with you some of the library independent work that we've done in my lab and Dr. George's lab as well. And let's see if I'm right with this. And in terms of outline for the presentation, we'll go through a little background, revisit some of the things that were covered yesterday, just as a point of refresher and a stepping off point. And then get into overview of some of the BST projects we're doing on Texas, focusing on library independent work, and then focusing on 3 specific studies. One looking at characterization of watersheds. The second, Joy Truesdale from Dr. Giovanni's lab will talk about evaluation and development of a feral hog marker they're working on. And then lastly, I'll wrap up with a project where we looked at using library independent BST to evaluate grazing management practices. So first, as speakers mentioned yesterday, we can break the the BST into 2 different main categories, either library dependent or library independent. And with the library dependent, specifically in Texas, what we're usually doing is taking the water sample, isolating E. coli, using EPA method 1603, membrane filtration on a modified MTEC auger, DNA fingerprinting those isolates and comparing it to that Texas E. coli BST library that Elizabeth talked about yesterday afternoon. Now, in contrast with library independent BST, the big difference being the elimination of the need to develop that known source library that can be very expensive and time consuming. And for this, we'll take our water sample and we'll pass it through typically a 0.2 micron soup ore filter, run 25 to 100 mils of water through that, collect the biomass, extract the DNA, and then we're PCR amplifying for target sequences, and we'll talk about some of those on the next slide. And then we're looking at 2 different ways for the analysis. Either a qualitative presence-absence type approach, the marker's there, it's not there, or a quantitative approach. Now, even though I say quantitative, we're not necessarily looking for an absolute quantitation. This is more like what Dr. Shanks mentioned yesterday for BMP evaluations, that we're looking for a relative change typically with a BMP evaluation essentially. And again, the big advantages of the library independent approach being hopefully less cost because you eliminate the need to build a library and the samples can be processed more quickly. So in terms of the specific methods we're using, we're primarily using Bacteroidales or Bacteroides markers and primers. And as several of the speakers mentioned yesterday, these are organisms that are very common in the intestines of humans and animals. Actually, they're more numerous than E. coli in most of those environments. There's a lot of different markers out there for different species or different organisms. Some of these are more refined and more optimized than others. Usually some of these are very, very specific, although you could debate whether they ever reach 100% criterion or specificity. Now, one of the downsides for it is that there are relatively limited numbers of wildlife-specific markers. And this can be a big consideration in rural watersheds, but potentially even in urban watersheds. Somebody brought up the issue of urban wildlife yesterday. So that could be a potential limitation for these library-independent approaches. And then finally, as some of the speakers mentioned yesterday, the relationship between these markers and E. coli and other regulatory organisms and pathogens isn't fully elucidated. So in terms of Texas specifically, and where we're applying library-independent BST, and these are ongoing projects typically, we have 6 projects where we're characterizing watersheds. Lake Granbury and Buck Creek was mentioned yesterday by Dr. Hagedorn. Also, the Little Brazos River Tributaries project is the one that I'm involved with, that is the farthest along, and that's the one I'll highlight some of the results on here in the next subsequent slides. Also, we're doing a couple projects in East Texas and Big Cypress and the Toyac Bayou, and one a little south of here, the Leona River, that started up relatively recently. Additionally, we're looking at some best management practice evaluations, and this is focusing on edge-of-field runoff samples. And Dr. Giovanni's done some work looking at dairy manure application and primarily vegetative filter strips and the impacts that has on reducing bacteria runoff from edge-of-field samples in response to dairy manure application. And then also we have a grazing system study, which will be the final project that I'll talk about this morning. And then Dr. Giovanni has also investigated a waterborne disease outbreak in Oklahoma City and using the library-independent approaches to try to determine the sources of that. So to start in on the first study I was going to focus in on, this being the Little Brazos River Tributaries project, this is a Tier 2 BST approach. So if you remember back to Aaron's presentation yesterday, this involved a toolbox type approach, and is recommended by the Bacteria Task Force, involved a toolbox type approach where we do limited library-dependent BST in conjunction with library-independent BST, and also potentially other things such as modeling and other approaches that were involved in this. Now for the library dependent, the limited library dependent, what we did was analyze E. coli from 81 water samples across the watershed, and I'll show an illustration of the watershed here in the next slide, the subsequent slide, and then fingerprinted those using the ERIC RP and the rapid printing approach, and then matched those back against the Texas E. coli BST library that Elizabeth covered yesterday for the best match and the source identification. For the library interdependent approach, we analyzed 259 water samples across the study area using various Bacteroidales markers. And again, this is a presence-absence type approach. And the markers we used were human, ruminant, which would be cattle and deer and other ruminants, hog, and the horse markers. And to show the highlight of the watershed, and Dr. Karthikino will go in more detail on this later on when he talks about the modeling for this project. But it was a relatively rural watershed, which is an important point, only 3 small towns in there and relatively homogeneous. Additionally, there were 5 sub-watersheds. So there are 5 individual creeks that fed down into Little Brazos River. So we had Walnut Creek, Mud Creek, Pin Oak Creek, Spring Creek, and Campbell's Creek. And we had 2 sampling locations as indicated by the little blue circles on each sub-watershed where we did sampling for each site. So we had a total of 10 sampling sites that were present there. So in terms of the samples that were collected, we collected over about a 1-year period going from May of 2009 to April of 2010. And we segregated the samples relatively evenly between normal flow conditions and storm flow conditions. So we had both the Bacteroidales samples and the E. coli samples. And specifically for the Bacteroidales, for the normal flow, we had 130 samples we analyzed, and under storm flow, 100 samples. And likewise on E. coli, 35 under normal flow and 35 under storm flow. So relatively even distribution between normal flow and storm flow conditions. So to get into the results, and first we'll look at this as a sub-watershed split. So break them all out by the individual sub-watersheds. And again, this is presence-absence. So the values on the y-axis are percent incidence essentially. So what percentage of the samples tested positive for each one of these markers. And then for each of the markers, we have the human marker in red, ruminant marker in blue, hog marker in green, and the horse marker in purple. So probably one of the first things that jumps out is there isn't a whole lot of variation looking across those different sub-watersheds. And this isn't real surprising. Like I mentioned, this is a relatively rural watershed. It isn't a real complex source of different inputs that are there. So this at least makes sense. And that's one of the, I guess, the good things for the outcome is when it makes sense with what you know about the watershed. The second point is in terms of the relative incidence of the different sources, with the hog marker being most frequently detected, followed by the ruminant, and lower proportions of both the human and the horse heads. So when we sum this up, and it's that same data, just compiled across the entire watershed, graph set up the same way, that the hog marker was detected in 71% of the samples, the ruminant marker in 39% of the samples, and both the human and the horse in less than 10% of the samples. So how does this compare to the limited library-dependent BST we did as part of that toolbox approach to hopefully complement some of the limitations of each of the methods? So I'll break this out in a 3-way source classification. In the red, we have the human sources. In blue, domestic animals, and this would include livestock and pets and everything rolled into that. Green, wildlife, and then the white, the unidentified isolates. So it matches up relatively well with the library interdependent approach. Relatively small proportion of human isolates, essentially, a larger proportion of domestic animals, including livestock, but the vast majority, or the largest majority of samples being from wildlife. Now, one thing I should point out here is that we did have a few of these isolates that came back. Their closest match was to E. coli from a hog isolate in the library. And based on what was known about the site, there wasn't much domestic hog production, so we attributed this as likely arising from feral hogs. So we moved it from the domestic hog, or at least the domestic animal category, into the wildlife category. But that was a relatively small number of the E. coli isolates. Okay. So wildlife being the biggest contributor, at least source classification, based on this limited library-dependent BST. Now, one other thing to point out here is that 16% unidentified. So that means that we were able to identify, using the existing Texas E. coli BST library that Elizabeth talked about yesterday, 84% of the isolates. And this will be important when we talk about the conclusions here in a minute. Now, how about breaking out by storm versus normal flow? And the graph is set up the same way here with the same markers under base flow conditions versus storm flow. And you see the relative level of the different markers, the relative contribution isn't changing much. The major difference here is maybe a higher proportion of samples testing positive for ruminant and hog markers, but still you see the relative ranking being the same between either storm flow or normal flow conditions. And very similarly for the limited library-dependent approach, again, there isn't a change under storm flow conditions between the relative ranking of the categories, albeit there may be a slightly higher proportion of samples identified as wildlife under storm flow conditions, but relatively comparable results, at least based on source classification, under regular flow and storm flow conditions. So in summary on this project, focusing on the BST, with the limited library-dependent analysis, if you remember back to Aaron's talk yesterday, one of the numbers that could be used based on the task force report to evaluate how well the Tier 2 approach is working is whether we're able to identify 80% of the isolates using the existing library. If that isn't the case, we may need to go back and add in isolates from the specific watershed we're doing work on. So at least based on that criterion, it looks like it's working pretty well. We were able to identify 84% of the isolates. Now with that said, as subsequent projects, we actually have went back and collected known source isolates, some from Dr. Karthikeyan and other collaborators, and we fingerprinted over 80 different known source isolates that we sent to Dr. Giovanni. They're going to put in the library, and we'll repeat the analysis and hopefully even further refine and eliminate some of these non-identified isolates. But with the current analysis, it appears that the major water isolates in the watershed appear to be wildlife, and this can include feral hogs, deer, avian wildlife, and other small mammals, with smaller proportions of domestic animals, including livestock and pets. And similarly with the library independent analysis, the hog marker was detected most frequently, 71% of the samples, followed up by the ruminant marker. And I should make the point that, again, based on that source survey, there wasn't much hog production in the watershed, so we assume that this hog marker is hitting primarily feral hogs is kind of the hypothesis going forward. And there's a small proportion of hits for human and horse markers. So eventually we take these results and combine them with other tools in the toolbox. So including the land use, a watershed source survey, and the modeling. And Dr. Karthikeyan will talk about his modeling results for these watersheds here later this morning and how these BST results either agree or they don't agree, and if they don't, how we should go forward on that and try to reconcile some of it. And then we'll take this information, share it with the stakeholders to aid them in the watershed protection planning process. Now, if this watershed specifically and the other ones where we presented similar type of results, where wildlife has been a major component in terms of contribution, and especially when that hog marker lights up like that, the stakeholders typically grab onto that because there isn't a whole lot of love lost for feral hogs in the state of Texas right now. Any excuse to be able to shoot them from helicopters is grabbed onto. So that's the reason why Joy is going to talk about this next segment where they're working on developing a feral hog marker and be able to better differentiate those out. So, Joy.
Thank you, Dr. Gentry. Like Dr. Gentry said, I'm a student with Dr. Giovanni in El Paso, and I'm going to give a very brief overview of some of the research that we've been doing with feral hogs. Landowners face many challenges as they manage their land for agricultural uses as well as for wildlife habitat. But one of the major hindrances that landowners face is invasive species, and one of those invasive species is feral hogs. Now, most people think that feral hogs are just kind of in the southwestern part of the United States, but studies have now shown that feral hogs have been seen in the southeastern states, Michigan, Iowa, Nebraska, New York, Pennsylvania, Wisconsin, and recently they even observed feral hogs in Hawaii. Now, for Texas, it's a major issue because we have a population of nearly 2 million feral hogs, and they inhabit bottomlands such as rivers, creeks, and drainages. And because of this, we have concerns over their impact on water quality. Also, they compete directly with game, non-game, and livestock for food, and they also destroy native plants. And another reason there's such a problem is because they cause about $52 million in damage in Texas alone every year. I would just like to mention, does anybody know that there's a university, a major university in El Paso, Texas? And it's called the University of Texas at El Paso, and it's also referred to as UTEP, or Texas College of Mines. I just wanted to throw that out there. I digress. We did use a modified PF163 assay to see if it would determine, differentiate or detect domestic pig as well as feral hog. And so we did test various samples from these different locations, and we found that about 100% of the time the marker did detect domestic pig as well as feral hog. We then use qPCR and high-resolution melt analysis to look at these, to characterize these PCR products. With your standard melt curve, you'll get something that looks like this. When we use the high-resolution melt analysis here, we get more information on the melts of the amplicons from the PCR products. Using conventional PCR, we get, we run just the standard agarose gel, and it's just a presence-absent test to see if there's pollution in the water. And then we use our high-resolution melt analysis to get more information, and we're currently sending out these PCR products to characterize them using sequence analysis to see if we can see any difference between the domestic pig and feral hog amplicons.
So the last project I want to share with you is looking at a library-independent approach to evaluate the impacts of grazing management on bacteria runoff, specifically E. coli and some Bacteroidales markers specifically we're looking at. And this is Kevin Wagner's research. So for this, we looked at 3 different treatments at 7 total sites, a no grazing treatment at 3 locations, a moderately stocked, which would be recommended grazing rates at 3 locations, and then a heavy stocked, which would be a 2x grazing rate at 1 location. And specifically for the locations, the first was the Welder Wildlife Refuge at Sinton, and this is down in South Texas near Corpus Christi. As you can see by the picture here, it's not necessarily what you think of as a normal pasture type situation. It's a mixed chaparral with a lot of woody vegetation present on the site. The second site was at Riesel, which is up near Waco, and this included both a native prairie grass site and a Bermuda grass pasture or graze site. And the prairie grass site is kind of unique in that the last time that it was grazed was at least early 1900s essentially, so it's not been grazed for a long time. And then the final site was the Texas A&M Beef Center near College Station, and invariably I'm going to refer to that as Brazos Bottom later on. So if I say Brazos Bottom and Beef Center or College Station, those are all interchangeable essentially. And it was planted in Tifton 85 Bermuda grass. Now from these sites, we collected edge of field runoff with automated samplers as and if rainfall events occurred, which has been somewhat of a challenge over the past few years at some of the locations. We enumerated E. coli using the EPA method 1603, so the membrane filtration modified MTEC approach, and then also assayed Bacteroidales using Alice Layton at the University of Tennessee's primers. And the 2 we specifically used, and this is a qPCR TACMAN type approach, the 2 primers we specifically used were the total Bacteroidales, or the all-back, and the bovine associated, or the bowback. Now, at least a couple of speakers yesterday, I think Dr. Steckel and Dr. Shanks mentioned bowback and some of the issues that they've seen with cross-hybridization and non-specific detection. Hopefully some of that will come out in the slides we talk about here later on, but we did see similar things with the bowback at least. So to start off with a big summary of the data, and this is a pretty involved table, and it breaks down the data by site by year. But what we want to do is kind of break this up into smaller segments, and we'll kind of walk through it a little bit. The take-home message that I'm trying to drive from this is that there wasn't a consistent trend with the behavior of the markers across sites over time. So let's look at some of these parts of it. First in the highlighted section here, and I'm not sure if you can read that from the back or not, but basically this is the Brazos Bottom or the College Station site. In 2009, BB1 no grazing, BB2 regular grazing, BB3 heavy grazing, and looking at all back median concentrations, and this is copy number per liter of runoff water, and then versus the bowback, which would be the bovine associated ones. So essentially with increasing grazing, if anything, you could say it's decreasing, which is counterintuitive. And especially with the bowback, there isn't a consistent trend with grazing treatment there. Additionally, when you compare across this at the 2X grazing, there's nearly a 3 order of magnitude difference. So that's really different than what I would expect in terms of the results. I'd expect a much more significant or higher contribution of cattle being detected by the bovine-associated marker. Now, in contrast, when looking at the exact same sites in 2010, a slightly increasing trend with an increasing grazing, but a much higher level of bowback, and especially when looking at the overgrazed site, it's relatively comparable, only about an order of magnitude difference between the bowback and the allback. So it behaved a little more typical, at least in 2010 at this one site. Now, one thing I should point out here, and this will come out probably on subsequent slides, at least with this one ungrazed site, there was at least one incidence where the cows didn't honor the fence and got across the fence, and they were on the site when there was a runoff event. So the marker did a really good job detecting that one runoff event when there was accidental grazing on the site. Now with Riesel, it did behave in both years a little more like you would suspect, with the SW12 being that prairie grass site, and this is in 2008, SW17, the normal stock site, with higher levels of allback and dramatically increased relative levels of bowback, or the bovine, in association with grazing. Similar study again in 2009, where you see over a 3-log increase in bowback in response to the grazing. So at some sites it tended to behave the way you'd think it would, and other sites it didn't. There wasn't a consistent trend. In Welder there wasn't a trend, but that's more complicated due to very limited runoff events down at Sinton. So how does this correlate with the E. coli data? And this slide and the next 4 graphs I'll throw up here are all correlating E. coli with either allback or bowback. And this one is at the Brazos bottom site near College Station, looking at, excuse me, wrong way, the allback concentrations on the x-axis versus E. coli concentration on the y-axis, and then the correlation for the non-grazed, moderately grazed, and heavily grazed site. You see a reasonable correlation there for those. R-squared value is 0.3, 0.4 or so. So a reasonably decent correlation between those 2 indicators at that one location. Now with the bowback, it started to kind of fall apart a little bit, at least the association. With a couple of the treatments, the ungrazed and heavily grazed, you could really stretch it and say there probably is a decent correlation, but surprisingly with the BB2, there wasn't. Okay. So the bowback didn't perform nearly as well there. Okay, how about the other 2 sites? Well, I'll just show you the Riesel, because the Welder is similar on it. Is that with Riesel, for neither marker, there was a little to no correlation between E. coli and either the allback or the bowback. So again, very different results depending on the watershed that we're looking at. So let's dig a little deeper and look at the E. coli data and split this out by watershed and by year. And focus out here first on just the portion here highlighted in the blue rectangle. On the y-axis, we've got E. coli concentration and colony forming units per 100 mils of runoff. And then we got our different sites by year again. And just to make it easier for illustration, the green bars indicate no grazing treatment, the red bars are normal grazing, and the blue bars are heavy grazing or overgrazing essentially. But looking across here, even though there appears to be a higher level at the ungrazed site in 2009, there isn't any significant difference or response to grazing treatment to E. coli concentrations in runoff for any of those years at those sites, which is kind of surprising to me, especially when you think about 2x grazing rate or overgrazing and not seeing higher levels of E. coli coming off in the runoff. Okay, so another case of it doesn't necessarily follow the trend you'd expect. Now, when we look over at the Riesel site, it was a different story. So this again, we're comparing the green, that non-grazed prairie site, versus the red, the typical grazing rate. In both 2008 and 2009, grazing led to higher levels or concentrations of E. coli in the runoff. So the story is pretty clear. It's matching up with, at least intuitively, I would expect. However, when you look at 2010, it falls apart and it goes the other direction. And if you're just looking at 2010 data, you can make the argument that grazing actually leads to lower runoff, so you could graze your sites and improve your water quality at least. Okay. But however, there's obviously something going on here temporally. And what Kevin did was went back and looked at this data a little more deeply to try to figure out what is happening with time to these sites to cause them to behave so differently over time. And what it turns out that happened was that in 2008 and 2009 at Riesel, it just so happened that every time there's a runoff event, cows were either on the site or they've been recently on the site. So these sites were all rotationally grazed in order to let the cows graze, let the grass recover, and put them back on. And it just so happened in 2008 and 2009 that every time there was a runoff event, cows were either on the site or they'd been on the site within the preceding 2 weeks. So he went back in and looked at the data a little differently. In addition to breaking it out by treatment where it's either ungrazed or grazed, he broke it out whether it was stocked or destocked. And stocked meaning that the cows were on the site when it rained, or they've been on the site in the preceding 2 weeks, and destocked being the opposite of that. And when you break it out that way, it really makes a lot of sense and really cleans up or clarifies some of the E. coli results at least. So we have the E. coli concentration on the y-axis. All the blue bars are destocked or no grazing treatments, and the green bars are the stock treatments. So some pretty obvious differences in response to stocking rate essentially. Now the question is, how does this work with the Bacteroides markers, our library-independent approach? And we'll show the worst performing of the two markers here, the Bobac. In this case, we got the Bobac concentration and copy numbers per liter on the y-axis, and these are box and whisker plots. So the solid line here is the median value, and the dashed line is the mean values here for each treatment. But in this case, there's actually 4 stock treatments essentially. The 3 at the top were the intentional stocks, and this is that one outlier where the cows got across the fence essentially. But you can see in all cases, in these stock sites, when you're breaking it out that way and segregating it, there's much higher levels of Bobac being present. And we're talking 2-3 orders of magnitude in most cases. Okay, so it worked pretty well on recently deposited fecal contamination. Now, another side, and I wanted to bring out this cross-hybridization issue, is that if we look at SW12, for example, this is that prairie site that hasn't been grazed in going on 100 years, essentially. Even at that site, seeing 10 to the 3, 10 to the 4 copies of this Bobac marker coming off in the runoff water per liter. So it's coming from somewhere. It's not coming from cows. It could be coming from deer. It could be coming from dogs. Who knows what? We don't know. But there's obviously a background contribution to that marker, a non-specificity. And you could imagine that once this water, if we're getting this level of background comes off and it starts mixing with other waters and surface water bodies or wherever, and you get this kind of mix of poo that was discussed yesterday, this high level of background detection could really wreak havoc with trying to delineate source contributions, essentially. So works well as long as you got a much higher cattle contribution, but as soon as that starts diminishing relative to non-specific detection, it may cause some major issues in source delineation. So how about taking back to the correlation and looking at the E. coli? And what this is, if you remember back to the first correlation slide I showed, that showed the Brazos bottom of the College Station site correlating E. coli with the Allback and the Bobac markers. And this is going back in and just pulling out that data when the sites were actively stocked, essentially. So we're refining that data set, looking at recent deposition. And in this case, we got the E. coli on the y-axis versus the different markers on the x-axis. And both with Allback and Bobac for that College Station site, saw really strong correlations. And I'm pretty excited about this, given the size of the watersheds, there's multiple hectares, and these are biological indicators which are notoriously don't behave the way you expect. So this is a pretty strong correlation, at least in my mind. So it really helped clean things up and worked well for this site. Now with that said, didn't help so much for the Riesel site. So it even helped further with one, but not the other sites. And the last data slide I'll show, and this was an attempt to try to get at using the Bobac, Allback markers to estimate a relative contribution from cattle to fecal or Bacteroides markers, essentially. Now, unlike the first study I talked about, we didn't do E. coli-based BST, so we didn't have a point or a cleaner point of comparison. So the way we did this was look at E. coli levels when the sites were stocked versus when they were destocked to try to estimate a cattle contribution, and then compare that with the Bobac copy numbers versus the Allback copy numbers under stocking rates or destocking. And the values I just want to point out here, and this is percentage estimated to arise from cattle. The hatched being the E. coli and the solid bars being the Bobac, Allback markers. The 3 highest values here that all hover around 60% are the actual stock sites. So that makes sense. There's more cattle contribution probably when the cows are there than when they aren't. But the difference here being, by using that E. coli estimation, the average value is around 60, plus or minus a great deal. As you can see with the error bars, there's a lot of variation that was there. Now in comparison, the Bacteroidales or Bacteroides-based estimation was lower, averaging a little over 20% or around 20% at each one of the stock sites. So we can't draw some really clean conclusions based on the variability and the issues we have with E. coli in the way we're doing the calculation here, but it definitely suggests at least that the bowback-based estimation is underestimating cattle contributions. So definitely, and even intuitively, if the cows are on the site, I would assume that they're probably contributing more than 20% of the loading to that field usually. Even though there's wildlife there, there's probably a minor proportion compared to the cattle loading essentially. So in summary of that, both the markers in the runoff were higher in the runoff when they were actively stocked. And this suggests that they could be good markers for recent deposition. Now, the difficulty arises when segregating recent from long-term, and undoubtedly this is due to differential die-off of E. coli versus some of the markers and various other complications on it. Additionally, the bowback-allback ratio is generally aligned well with stocking rate, and what this means is that when they were stocked, you saw a higher proportion arising from bowback. However, it appeared that that approach underestimated or likely underestimated bovine-associated contributions. And then finally, very obvious different results from the different watersheds. Due to geographic variability in the markers potentially, it worked really well, especially when we looked at recent contamination in the one watershed. And one point I should make from that is the standard curve was developed from cattle feces or cattle manure sample from that watershed where it performed best. So at least there's a possibility that if we developed our markers from those other watersheds or developed watershed-specific markers that would improve those relationships. And we're going back in there and we're doing some of that to try to evaluate it and see if we can improve it further. But that's at least a possibility. And finally, Meatloaf had a song a few years ago, 2 out of 3 ain't bad. Anybody remember that? So I'm not going to try to argue that 1 out of 3 ain't bad, but to try to look at it on an optimistic point of view, one thing, trying to correlate E. coli with this marker maybe isn't a fair point of comparison for it. And secondly, at least from my perspective and the limited optimism I still have from being in academia, is that taking a marker that Dr. Layton developed from Tennessee, primarily with isolates from the eastern part of the U.S., and a relatively small number compared to more recent studies. And I think there's only one manure sample from Texas, and there's like a longhorn from up around Dallas or somewhere. And taking that and it working well, and quite well, at least in one watershed, is encouraging, at least to me. And it seems like it's something to build on. And undoubtedly, as Dr. Shanks and Dr. Harwood and Dr. Sadowski and others develop these new markers, hopefully it can further improve this and build on it. And whether or not they ever get to that one smoking gun is another question, but I think they can all contribute to it and help improve our source delineations, essentially. All right. So as I mentioned, we're working on looking at the geographic variability. Joy talked about the feral hog marker. I have a PhD student, Emily Martin, who's trying to develop a deer marker to discriminate between deer and other ruminants, especially cattle. And she's using the high-throughput sequencing approach, kind of similar to what Dr. Sadowski outlined yesterday. And then a lot of these watersheds, especially in East Texas that we work in, have a lot of poultry, specifically chickens. And the stakeholders typically ask, well, what about the chickens? And to date, we haven't been looking at a library-independent marker, and we're really excited about potentially using Dr. Harwood's marker to delineate different source contributions from poultry in those watersheds. Now, with that said, one last point I'd make is that, and this may be more a question or something for the different speakers, but maybe later on in discussion or side discussions, is what suggestions y'all have for us. So if we take like Dr. Harwood's or a new marker and we try to bring it into Texas, what approach should we use for validation in order for us to have confidence that those results are going to adequately translate to Texas watersheds and Texas sites? So with y'all's expertise, if y'all have any suggestions, maybe later on or even after the meeting, I'd love to talk with you, and I'm sure George would as well. Okay. And just acknowledge the people that actually did the work. Joy, you heard from earlier, and Elizabeth yesterday, Emily Martin from my lab, and Kevin Wagner from Texas Water Resources Institute, and the Texas State Soil and Water Conservation Board, who funded the vast majority of this work. And with that, I'd be glad to try to answer any questions if we have time, or else I'm around so I can do it later.